Hi there, and welcome to the second lecture for the second chapter of Nature in Code. Today I want to talk about the Hardy-Weinberg principle. What is the Hardy-Weinberg principle and why is it important? Well, I'll explain to you in a minute what the Hardy-Weinberg principle is, but let me just tell you why it is important. It is important because the Hardy-Weinberg principle is the null model of evolutionary biology, at least from the perspective of genes. It is the null model. What is a null model? A null model kind of tells you what to expect. And that's really important if you think about it, especially in science. Um, as you do research, you find a lot of things that are expected, and those aren't typically that interesting because, well, they were expected. So they're based on knowledge that we already have. It's typically when you find something that is completely unexpected where people go, that is interesting. And if you actually look at the history of scientific breakthroughs, those are all unexpected things that suddenly popped up. So in genetics, in evolutionary genetics, the null model is the Hardy-Weinberg principle. And let me just write that again here. Hardy-Weinberg principle. The Hardy-Weinberg principle was described by two mathematicians called Hardy and Weinberg about 100 years ago. It's actually a fairly simple idea, and Hardy and Weinberg themselves were surprised that this wasn't more widely known in biology back in the day. So what is the idea? Well, the idea is as follows. First of all, some basic biology. We just talked about DNA in the last lecture. I just briefly mentioned that DNA is the molecule in which genetic information, which is the hereditary information, is stored. And all your genes are lined up on this strand of DNA. Now, it turns out that certainly almost all animals, or most animal species, are so-called diploid species. What does that mean? Well, it means that not only do they have one set of genes, but they actually have two copies of the genetic information in each single cell. That is opposed to some species which are haploid, which means they only have one set of genes, or there are others where some cells or even some species are triploid or even have more copies of genes than just one or two. But certainly for animals and most plants, diploid is most common. So diploid meaning two copies. Now, if you think about reproduction, so individuals are made up of cells, and each of these cells have two copies because they're diploid. Now, there's a process by which individuals produce so-called gametes. And gametes are cells specifically used in sexual reproduction. And what's special about these cells is that they are haploid. And they are produced by a process called meiosis, which you may recall from your days back in school in the biology class. It doesn't really matter how this works. But the important thing is all your cells are diploid except once your body engages in the process of meiosis and starts producing gametes, then those gametes, the cells specifically for sexual reproduction, only have one copy of your genes. And that, of course, makes a lot of sense because, say, now we have another individual over here, again, a set of cells, also engaging, of course, in the same meiotic process, creating gametes with just one set of genes. Now, if they fuse in the process of sexual reproduction, they produce a cell, the first cell, which is called a zygote, that now has 2 copies again. And so one copy, I'm just going to denote here in blue, from one individual, and in red from the other individual. Okay, so this is the blue copy here, simplified of course, and this is the red copy here. Now, based on this individual will now, of course, or the cell will go ahead and build a body by copying this cell again and again and again in the process of development. And then you have this individual down here, and the cells all have 2 copies, of course, again because they're diploid. But it's important to remember that one of those copies comes from one individual, here the blue one, and the other copy comes from the other individual. And that is true for you, dear listener, and for me, of course, and for everyone as well. I have half of my genes from my mother and half of my genes from my father. So let's now build on this model and reduce it to a simplified version. And let's just zoom in on one gene, okay, just on one gene. And that gene we could just call the A gene. And it comes in 2 versions, okay? And as we now know from last lecture, these versions are called alleles. So let's say we have the A1 allele and the A2 allele. So if you now think about cells, okay, this is a cell with 2 copies. The cell, or this individual, for which the cell is representative, could have an A1 here and an A1 over here. Alternatively, it could have an A2 here and also an A2 over here. So it could have 2 copies of the A2. Or the third possibility is that one of the copies would be A1 and one of the copies would be A2. Okay, so these are the 3 possible combinations: A1A1, A1A2, or A2A2. Now we call these cells, or this genotype, homozygous, okay, for the A1. And this guy over here would be homozygous for the A2. And this cell here, this genotype, is heterozygous because it has different versions, namely A1 and A2. So these 2 types are homozygous and this type is heterozygous. And that's language we'll need throughout the course. So if we now are if we would be looking at a population and we would be interested in genotype frequencies. And by the way, genotype is just a set of genes. So we have here 3 possible genotypes, okay? 3 possible genotypes in this population. There's going to be A1A1 genotypes, there's going to be A1A2 genotypes, and there's going to be A2A2 genotypes. So if you now look at a population, we would like to understand what is the distribution of genotypes in this population. What are the frequencies of genotypes? So say we are looking at a population, of 100 individuals, just to make the math a little easier. 100 individuals. We count them all up, we look at their genotypes, and let's say we find the A1 genotype in 15 individuals. And let's also say we find the heterozygous genotype A1A2 in 50 individuals, and we find the A2 genotype in 35 individuals. And that makes sense because that adds up to 100, and of course it has to. Okay, so the genotype frequencies here are 15%, 15 divided by 100, 50% for the heterozygote, and 35% for the A2A2 homozygote. So these are the genotype frequencies. Excellent. So now the next question is, well, what are the allele frequencies? And remember, evolution, the definition of evolution is a change of allele frequencies in a population over time. So we always want to know allele frequencies when we're concerned about understanding evolution. So we now know the genotype frequencies. What about the allele frequencies? Well, that's actually also very easy to find out. So keep in mind we have 100 individuals, okay, and those are diploid individuals. 100 diploid individuals. Each individual has 2 sets of genes, right? So we expect 200 copies of the allele. 200 copies of the alleles. Okay, because we have 100 diploid individuals. Now let's take a look at those 15A1A1 individuals. So what about them? Well, how many A1 alleles do they provide? Well, each of them has 2. So they provide a total of 30 A1 allele copies. We said we found 50 A1, A2 individuals. So these 50 genotypes, how many A1 alleles do they provide? Just think about this for a second. Okay, so they provide 50 A1 alleles. Why is that? Well, because they only have 1 copy of the A1 allele. The other copy is an A2 allele, which means we also have 50 A2 alleles that are provided by these genotypes. And then finally we have 35 genotypes of type A2A2. So each of them is providing 2 A2 alleles, which means we have a total of 70 A2 alleles provided by the A2A2 genotypes. And what that means is we have 30 plus 50, which means 80 A1 alleles, and we have 50 plus 70, so 120 A2 alleles. And that, of course, is as it should be, because those together should, of course, add up to the 200 alleles. All right, so now having the absolute numbers, we can calculate the frequencies. Okay, so the A1 allele frequency is 80 alleles out of 200, so 80 divided by 200 is 40%. And correspondingly, the A2 allele frequency is 120 alleles out of 200, so 120 divided by 200, so that is 60%. And again, because there are only 2 alleles, those 2 frequencies need to add up to 100%. Okay, so that's great. So we've now established in this population of 100 individuals, where we have a gene that has 2 versions of itself, namely A1 and A2, we have 15% A1A1 genotype, we have 50% A1A2 genotype, and we have 35% A2A2 genotype, and that translates into an A1 allele frequency of 40% and an A2 allele frequency of 60%. Okay, so that was relatively straightforward. Now, in the next video, we're going to look at what those allele frequencies will be in the next generation. And then this is where the Hardy-Weinberg principle will come in. All right, I'll see you in the next lecture.